Drew Endy is an engineer who's moved from MIT to the West Coast, is probably the most skillful proponent of biobricks. There's a foundation. There's an international competition aimed at students who are supposed to design the iGEM. They design these little biological machines, and then they compete to see who wins the prizes. The biobricks movement, at least, is it's attempting an open source model. So if you want to deposit a biobrick with the Biobricks Foundation, you are supposed to allow anyone to use it for any purpose, as long as they continue to make it an open source application. So it sees a distinct analogy to the early days of the computer movement and the open source software. Stream number 3 is some high-level genetic engineering. And this is the one I said that can't claim a certain amount of success. This is Jay Kiesling and colleagues have managed to create a system, a bug, that produces artemisinin. And not everyone's eyes, this is an unmitigated good, however. We'll visit why in a moment. Now, the key question that scientists are asking is synthetic biology really different from other means we have of hacking data? And they use this phrase, hacking biology, in the way that virus makers hack software. Or honest people just try to take existing software, modify it, and use it for somewhat different purposes. We've been able to do genetic modification since at least 1974, and the publication of the first paper describing the creation of the transgenic bacteria, the beginning of the recombinant DNA curve. Now, it is true that synthetic biology does have a different ethos that permeates it. It's not so much hypothesis-driven, although some streams are more so than others. But it's unlike most science, most biological science, being driven by hypothesis, the quest for understanding, and more by the quest to build something, to make a particular thing happen. One view of synthetic biology is illuminated in the title of a report that the ETC group did when they called it Extreme Genetic Engineering. Now, molecular biology, as it's been historically and continues to be practiced, is not quite the same as what I'd like to call your biomolecular engineering. For one thing, historically, biologists have been exposed to ethical debates over the aims and consequences of biological research for decades. You will know the ELSI program in the U.S. is the Ethical, Legal, and Social Issues Program. I actually proposed that at one of these notorious congressional hearings in 1988. It became a reality in 1990, and I know that the EC, I think, has had a comparable program. So biologists, particularly geneticists, have become accustomed to at least knowing that there are ethical dimensions to their work. And in the overwhelming majority of people, at least that I meet, they take that obligation seriously. Furthermore, engineers are very different from scientists generally. The culture, training, and mindset of engineers is, it's a whole different professional culture. Now, not to say that there isn't some merging and interaction among the cultures, but engineers and scientists come at problems in rather different ways, traditionally. We discovered this when we did a report for our National Institutes of Health on nanotechnology. And we talked to several dozen high-level practitioners of nanotechnology, and the scientists were very worried about what the engineers were doing. They said they really don't understand the ethical implications of their work. Well, maybe that's just bias on the part of scientists, but it alerted us that there is a two cultures problem in nanotechnology. There's a two cultures problem in synthetic biology, I think. Not the two cultures problem that C.P. Snow wrote about, but different cultures of investigators. So one way to compare the two is to say that molecular biologists are concerned with discovery, the engineers with design. Biologists with emergent properties. The engineers want control and prediction. The biologists want to understand complexity. The engineers want to simplify and streamline what we're dealing with. So Tom Knight said, an alternative to understanding complexity is to get rid of it. Would you guess which camp he's in? So that's all I'm going to say in the way of describing the different streams of the science. It's obviously a much richer terrain than I have time to deal with. But I wanted to now try one of these, introduce a framework for thinking about it. So look at the way that the US and the UK have dealt with two emerging technologies, embryonic stem cells on the one hand, genetically modified organisms on the other. Roughly speaking, the US has been politically hamstrung, for the most part, trying to deal with embryonic stem cells. We've just had a recent breakthrough with the most recent election. But even so, it still remains a very difficult political topic in our country. It seems that the UK has done much better with it than we have. On the other hand, the US has had no problem, doesn't seem to raise any issues about GMOs. They're broadly accepted widely in the US food supply, whereas that's been a much more difficult problem over here. So, as I say, things have changed. The President issued an executive order in March, and just a week and a half ago, NIH issued its draft guidance on embryonic stem cell research. That pertains only to what research will be eligible for funding by our national institutes of health. That's the only regulation of it. You may or may not be aware that trying to actually clone a human baby is not against the law in my country. And trust me, it's not because people think it's a good idea. It's because you cannot have that conversation at the national legislature. I gave a lecture at Princeton University years ago, and somehow the question came up, why is it that the U.S. Congress hasn't been able to pass a law for about, probably it was about a month? I said, well, my view is that if you try to say anything about embryos in the U.S., you are going to alienate one of two parties. You're going to alienate right-to-life groups because they regard the embryo as a person and therefore sacred. If you try to permit cloning for research, somatic cell-nuclear transfer, say to create stem cell lines, in their view, what you're doing is you're killing a baby. If you pass a law that permits it for research, but does not prohibit it for reproduction, then they say, oh, so I can make a baby only if I promise to kill it? That's how it's seen. On the other side, you have the infertile couples who are just as passionate in many cases about wanting access to infertility treatment. And so you're either going to, if you restrict it, work with embryos, you're going to anger infertile couples. If you permit it, you're going to anger right-to-life groups. This is called a classic lose-lose proposition for a politician. So I chanced this remark, and then a member of the audience stand up and said, well, I'm General Counsel of the Department of Health and Human Services. This is a cringe moment. And she said, you're right. It hasn't happened before, so it was a very nice moment. In the UK, embryonic stem cell research has been handled, whether or not you're totally satisfied with the way it's been handled, it's been handled rather openly. It's been handled by an accountable political process. And many of us in the U.S. are envious about the way we've been able to reach principled decisions about the use of